Reporting live from Monkeytown, West Virginia, we have a fiery debate between famous scientists Belina Nye and Shelly. Is CRISPR technology ethical? Would interspecies families ruin the human race?
Hi, my name is Shelly. Ever since I was a wiggly little sperm, I knew my sole purpose in life was to give birth to a monkey. I love monkeys, and I think my life would be complete if I gave birth to a monkey. And I think not only would it benefit me, it would benefit the entire human race.
Now, here to talk to us a little bit about CRISPR and if having a monkey child is actually viable is Dr. Belina Nye.
Hello, I am famous scientist Belina Nye. Do not ask about my correlation to Bill Nye, there is none.
So, is it actually possible?
Well, we have the means in place to do it, it's just not as far as we've gone yet. So it is possible, in theory. We just don't have what it takes to accomplish it. However, we do have a computer program that we have used to generate what a monkey child from Shelly may look like. And here are the images that it conjures. This is your future, Shelly.
Oh my god, I finally figured it out! I know how CRISPR works! Bacteria just use CRISPR as part of the immune system by cutting out viral DNA from bacteriophages. So, this system allows bacteria to effectively kill viral DNA so the virus can't infect the cell. I'm so smart!
So what is CRISPR? CRISPR stands for clustered regularly interspaced palindromic repeats. And in bacteria, it's a phenomenon of these repeating sequences of DNA, and the bacteria in between those sequences can keep a record of previous viral attacks. CRISPR also represents the technology that humans can use to cut and edit DNA. This is a model of CRISPR. First we have the CRISPR-associated protein, and next we have the sgRNA, or single guide RNA. The sgRNA is made up of 3 parts: the 20-nucleotide guiding region, the hairpin loops, and the scaffold region. The sgRNA binds to Cas9, and this complex runs down DNA and attaches downstream to the protospacer adjacent motif, or PAM. The PAM sequence goes from 5 to 3 NGG, where N can be any nucleotide. Then the enzyme cuts the DNA. When this happens, the cell senses the break in the DNA and prepares to fix it. At this point, we insert a donor or template DNA. Between the homology arms of the donor DNA, there's a sequence that we are adding to the gene. In this case, we are inserting a stop codon. On the DNA, ACT is complementary to UGA, which is a stop codon. So this is what the DNA will look like after it's fixed. When the gene goes through translation, the protein will not be fully formed because of the early stop codon.
Hi, I'm Dr. Mead, and this is
Jim!
and we're here to explain in vivo and ex vivo and how we can add CRISPR into the human body. So for in vivo, we do it inside of the human body. That sounds a little bit crazy, but what we do is we take a virus that will attack a specific type of cell, and we take out the viral DNA so it doesn't actually harm the person, and then we put in the DNA that we want it to have. Then that virus goes into the human body and attacks the cells that it's supposed to, and instead of putting the viral DNA in, it'll put the actual DNA and proteins that we want it to be inside of the cells.
And for ex vivo, we can take out stem cells in the human body, shock them with electricity so that there are holes in the stem cells, so then we can inject the donor DNA, the sgRNA, and the proteins such as Cas9. And then once these stem cells are fixed, we can put them and inject them back into the body. And for example, this is very effective for sickle cell anemia because these stem cells in bone marrow, they create red blood cells, so this can be very effective for that.
DNA can be used to create a stop codon that can be put into the exon region of DNA, and when it tries to express the gene or be copied, it can't express it once it's copied because the stop codon is there. To add a gene of interest or to knock in a gene, we add a donor template of DNA that has the correct improvised sequence of DNA for a specific gene. Once the DNA is cut, the repair enzymes will sense the break and use donor DNA to repair the DNA. The donor DNA is made of complementary sequences on both sides, but the middle near the cut includes another gene. When the DNA is repaired, the new gene is added to the sequence. This way, when the DNA is transcribed, there is a new gene and therefore a new protein to be translated.
There is a fine line between what is ethical or not. And Shelly, do you think birthing a monkey is ethical?
I think it's completely ethical. I mean, monkeys are already so close to, like, their genetic makeup is so close to what a human is. We're so closely related. I don't see how it could harm mankind in, like, humanity, you know?
But if we use CRISPR to mess with the genes of the monkeys, don't you think something wrong could happen? Like, it could cause the extinction of monkeys or something else?
I think rather than causing the extinction of monkeys, it would proliferate the species and just the population in general. It could make the species of monkeys more intelligent, and I think there could be a harmony and a cooperation between monkeys and humans, which is something that I think a lot of people would like to see.
And personally, I would like to see the proboscis monkey here. This would be purple. That's my take on the ethics. I really think with this proboscis monkey, we could have a new future.
I think that we as a society need to stop and ask ourselves, are we really ready for a new race of super monkeys?
Yeah.
It's been hundreds of years since CRISPR was first discovered. Now legalized, CRISPR is rampant, just like these perky purple primates here. The monkey child of ancient Shelly was patient zero in the CRISPR experiments for monkeys. Scientist Belina Nye was able to successfully use CRISPR to alter the monkey's vocal cord structure and change the color of the monkey's skin and hair. Now, we have talking purple monkeys that are arguably on the same level as humans.
Today I'm here with John Cook the monkey. It's John Cook the monkey at today's monkey protest for human rights. How are you feeling today, John Cook?
Hi everyone, my name's John Cook, and I'm a descendant from the first human child, Christopher. You might know him. He's a son of Shelly. You know Shelly. Anyways, I think monkeys are just as capable as humans in everything, so why can't they have the same rights as humans?
Yeah, I'm bananas for monkey rights.
Monkeys deserve rights!
Monkeys deserve human rights. First of all, us as primates, we are far more beautiful than humans. And second of all, we're more environmentally friendly. Monkeys consume 800 calories less than humans, and our diet consists mostly of fruits, okay? So, therefore, we should be the divine leaders of this nation.
So, Belina Nye, the 36th Junior, how do you feel about this protest? And how did this all come to be?
Hi, I am Belina Nye, the 36th Junior. My great-great-grandmother was able to utilize the CRISPR technology people found centuries ago to edit monkey genes so that monkey embryos would create monkeys of the same species but more advanced features. My family has also been able to advance the world with CRISPR. Look at the bananas here. It has been genetically modified to incorporate genes from every fruit in the world to create a super fruit. This fruit is almost magical, as it contains all the nutrients a monkey would need in one day. CRISPR has also been able to cure every disease on Earth, including cancer. People live for much longer and are much healthier now.
That's crazy. I support this protest too.
I'm a monkey from South Africa, and I support this message.